Hey everyone, welcome back. I think this is going to be our last video in chapter 5, so that's kind of cool, right? And in this video, we're going to talk about the human life cycle. We're going to focus on female and then male meiosis. So in females, meiosis is termed Ugenesis. Whoa. In males, this is termed spermatogenesis. So let's look at these. So in males, spermatogenesis is what produces sperm cells. And this occurs in the testes of males. So the testes are the sex organ for males. They are the primary sex organ for males. So this is where all sperm are produced. This process occurs, which I think I told you in the last video, but it occurs starting at puberty in males, and it continues until death of the male in general. We start with a primary spermatocyte. This is a diploid cell. So he is diploid, which you can see with that 2N. And we're going to replicate the DNA, and then we're going to go through 2 divisions. The first division, meiosis 1, produces 2 secondary spermatocytes, which are haploid but duplicated. Then we go through a second meiotic division. Each of those secondary spermatocytes goes through a second meiotic division, producing 4 spermatids that are haploid and not duplicated. So spermatids then have to maturate to become sperm. So a spermatid can't actually do anything. It's just basically a nucleus with half the DNA. So here you see the initial primary spermatocyte, okay? Primary spermatocyte undergoes its first meiotic division, producing secondary spermatocytes that are haploid. These end up going through a second meiosis, producing haploid spermatids. Spermatids then have to go through a maturation process. This maturation process helps them to produce that elongate tail that they need. Helps them to produce that acrosomal cap that will protect them, so that they can then go through the acidic environment of the vaginal canal. They can and survive, and they can swim all the way to the female egg, which is going to be, or the oocyte, which is going to be found within the fallopian tubes. So really quick, I'm going to go back one more thing. This process of maturation of sperm, that is called spermiogenesis. And that occurs in the epididymis. So the epididymis is the first part of the male duct system. So basically you have this testicle here, and then you have this epididymis that kind of covers a portion of that testicle. And then that is going to move on into the vas deferens, okay? So spermatids are produced inside. I'm just making little circles for my spermatids. They're going to move to the epididymis where they maturate, and they'll move. And as they get more mature, they move closer and closer to that ductus deferens, or the vas deferens. And then once they hit that vas deferens, they're now mature, they can swim, and they are protected. And then they can go on and fertilize an egg. So now let's look at female oogenesis. So oogenesis occurs throughout a female's life, but it starts before the female is born, during fetal development. So we produce all of the eggs that we will ever have. There's over 1 million eggs in each of our ovaries before we are born. All of these eggs go through the first part of meiosis. So all of the oocytes will go through and arrest in prophase 1. So they basically stop the division and they just sit there. And then we go through infancy, childhood, and until we hit puberty, those eggs are inactive. And most of those eggs, I won't say most, well, yeah, most, really, it is most. A huge proportion of the eggs actually degenerate. So by the time we hit puberty, we only have about 400,000 eggs left. So we started with over 1 million eggs. Now we have only about 400,000 eggs left. And every month we're going to activate some of those eggs. And one of those eggs is going to get special attention and will go through all of the rest of meiosis 1 and start entering meiosis 2. So at puberty, we have one special oocyte that goes through meiosis, the rest of meiosis 1. And then this cell, when it goes through meiosis 1, it produces a secondary oocyte. The secondary oocyte is, so our eggs are large. They have all the nutrients and everything, all the organelles and everything, and they have that large nucleus with half of the DNA, right? The secondary oocyte is going to be haploid with duplicated DNA. And with that, because we put all the energy in this egg, there's a tiny little polar body that is produced. The polar body is basically just, and I'll write it over here, it's a polar body. The polar body is just a bag containing the excess DNA. So a polar body is just a storage space that we can dump extra DNA, because we started with this diploid cell. We go through this meiosis 1, and now we produced a haploid duplicated cell, but all the nutrients, all the organelles are going to stay with this cell. We're not halving the entire structure. So just a little bit of cytoplasm is going to go in here and the extra DNA. And that's going to degenerate. So that polar body is just junk, okay? So now we have this haploid duplicated secondary oocyte that will then go through a second division, but only if it gets fertilized. So this oocyte can only go through the second meiosis if a sperm, a mature sperm, fertilizes the egg. If it doesn't get fertilized, that egg will never go through its second meiotic division. And in that case, it will degenerate as well, and it will then be released during the menstrual cycle. But if the egg is fertilized, then that egg, that secondary oocyte, finishes up. It goes through all of meiosis 2, producing a haploid cell with only one copy of each chromosome, so it's no longer duplicated, and it produces another polar body. That extra polar body, again, is garbage. Okay? And now we have this nucleus and we have the sperm nucleus. Those two will come together and this will form that diploid. That's a big chromosome, or that's a big nucleus compared to those two tiny nucleus, okay? And this zygote will undergo multiple mitotic, so mitosis, mitotic divisions and specializations, forming a new embryo, fetus, and then new human being. So let's look at that. Oh, okay, I'm sorry. Let's first look at this. So this is meiosis. We start with this primary oocyte that is diploid, okay? It goes through and interphase, replicates DNA, and then we go through the first meiotic, or the first meiosis. We arrest in prophase 1 once all of those eggs, once we are born. So basically during fetal development, we get to prophase 1 and all the eggs are arrested. Then we enter or finish prophase 1 after we hit puberty. And if fertilization occurs like this, then we go through prophase 2. And so here we have I'm sorry, I said that wrong. If fertilization occurs, then we go through meiosis 2, not prophase 2, meiosis 2. And so here we have a sperm fertilizing the egg. So this egg will then go through meiosis 2. Here you see that first polar body from the first meiotic division. Here's the second polar body from the second meiotic division. This sperm nucleus here and this egg nucleus here are going to come together, forming the zygote nucleus. The two polar bodies will degenerate and just be resorbed by the egg. And now we have this beautiful zygote that will go through multiple mitotic divisions. And so this is just both of those pictures together. And here is the full life cycle. So we all started out as this diploid zygote, right? The diploid zygote produces that unique human being. The unique human being can be male or female and will grow up into an adult. So male and female come together. They form their own sex cells, either sperm or egg. And the sperm and egg can come together, fertilizing. The sperm fertilize the egg, and we produce another new zygote. Okay, so I hope you enjoyed this chapter. I will be uploading all of this and we'll be getting into genetics next. So have a great day. Bye.